Hello friends, I am Ayantika and in this video I will be discussing about slime molds. This is the part 2 of the video where I will be discussing about the life cycle of slime molds. Now slime molds, as we know, that they were considered part of fungi by many researchers like Alexopoulos and Mims, but now after development of PCR it was put in another paraphyletic group of protists. This protists group included both animal-like protists, fungi-like protists, and plant-like protists. So today we will be discussing about the fungi-like protists, that is the slime molds, which was earlier included in Myxomycotina. And in this video I will be discussing about the life cycle. In the previous video, that of the slime molds, I have discussed an overview, that is a general characteristic of slime molds, their ecological importance, their research importance, their classification, their habitat and distribution. So here I will be discussing about the life cycle. So this video will follow the fungi series that I'll be starting. So this two videos will be about Myxomycotina that were earlier considered fungi. Of slime molds. Now the life cycle of slime mold, it is holocarpic, which means that the somatic phase is succeeded by the reproductive phase, which is again the reproductive phase is again succeeded by somatic phase. Both the somatic phase and the reproductive phase do not occur together at a time. So this type of life cycle, where a life cycle is divided into somatic and reproductive phase, that is they have two phases which do not occur together, this is called holocarpic life cycle, where organisms where the reproductive as well as the vegetative phase they occur together, they are called eucarpic. But our slime mold is holocarpic. So first we will be discussing about the somatic phase, which is divided into plasmodium and pseudoplasmodium. So there are two types in somatic phase. The plasmodium is multinucleated protoplasmic mass, and the pseudoplasmodium, it is also multinucleated, but the single amoeba, they retain their identity because they do not undergo fusion of nucleus. Whereas in the plasmodium is formed by syngamy, that is by the fusion of two type of nuclei, and it is diploid in nature. But on the other hand, this pseudoplasmodium, it is formed by the fusion of mixed amoeba that maintain their identity, and their nuclei do not fuse, and it is haploid in nature. So during the time of sporulation, these haploid nuclei, they of different kind, they just form different type of spores. But when it comes to the plasmodium, then the diploid nuclei undergoes meiosis to form spores, but during the meiosis, different type of genetic recombination causes different number of different kinds of spores. So the plasmodium, it is diploid nucleus, and the pseudoplasmodium, it is haploid in nature. The pseudoplasmodium, it is this can be observed by the fact that both the mixed amoeba, pseudoplasmodium and the plasmodium, they might look similar, but in their developmental potential they are bit different. Then this has been observed difference in the developmental potential of amoeba and the plasmodium is the mechanism of mitosis. Now, the plasmodia, as it has diploid nuclei, where the diploid nuclei is formed by the fusion of different the nuclei of different amoeba. So this plasmodium has open mitosis. As it has open mitosis, which means that the nuclear membrane undergoes breakdown before the division actually starts. But when we come to the pseudoplasmodium or the mixed amoeba form, they have closed mitosis, that is, cell division occur but nuclear membrane do not break down. So this type of close mitosis is to protect their identity, that is, maintain their individual identity. That is, if there is a breakdown of nuclear membrane, then it is very easy to for the fusion of the cells. So when they undergo division, then also they retain their nuclear membrane. And nuclear after only after the division do the single nuclei, they divide into two, but this division occurs with the nuclear membrane intact. And this intactness of the nuclear membrane is only to provide the sole purpose of this close mitosis is to protect the identity of the nucleus in the pseudo. So, in the somatic phase, we see the plasmodium. It is two types. One is the pseudoplasmodium and another is the plasmodium, depending upon its ploidy level. And but when we see about the structure, the plasmodium can be of three types. The protoplasmodium, which is the simplest, and it is uninucleate, it is very tiny, and it is also very slimy. So as it is very tiny, so they have a single sporangia, and the cytoplasmic streaming, that is of much research importance. Cytoplasmic streaming is very slow and very indistinct in the protoplasmodium. And then is the aphanoplasmodium, where the plasmodium is multinucleate and they have a delicate strand or a strong strand of network, and the protoplasmic streaming is a bit rampant than that of the protoplasmodium. So next higher level is the phaneroplasmodium, which is the phaneroplasmodium in its initial stage, it represents the protoplasmodium, but gradually it increases in size, and now this phaneroplasmodium is the type of plasmodium that is studied in most labs, because phaneroplasmodium is of enormous size. It has an anterior fan-shaped structure, and the posterior is a network-like structure. So this phaneroplasmodium is the largest of the three type of plasmodium, and it is the most widely studied of all the plasmodium. This division of the plasmodium was done by Alexopoulos and based on the structure, the size of the plasmodium. So this phaneroplasmodium, if it faces stress, like for example desiccation, that is rise in temperature, or desiccation, that is lower amount of moisture, then this plasmodium is converted into sclerotium. And the sclerotium, they have thick walls. Usually the plasmodium or various type of plasmodium do not have a cell wall but have a membrane. But when the sclerotia is formed, this membrane it thickens, and it is able to survive for years, and it is reactivated. The sclerotium, it gets reactivated only when the condition for survival is conducive, that is it faces moisture and the temperature is also low. So this three form of plasmodium divided by electropolis, and this sclerotium is is formed only when the plasmodium, it suffers from stress both in temperature as well as in moisture content. So that was all about the somatic phase. Now, when there is reduction in the amount of food or water, then also it undergoes sporulation. And actually, if there is reduction in the amount of food and water, and they have a huge amount of stored food material, then they undergo sporulation. And the sporulation or spore formation is generally two types. One is the exosporous, another is the endosporous. As the name suggests, exosporous, that is when the spore, they are not covered by any membrane, that is called exosporous. So sporulation, it occurs when the plasmodium, this is, say for example, this is a plasmodium, it develops a stalk-like structure, and then the spore is formed on the top of the stalk-like structure. This is called the sporophore. This is called the sporophore. I've written SP, and this is the main sporangia, where spores are formed, and the when the spores are formed, they can be covered by a membrane, so they are endosporas, or they can be devoid of any membrane in their matured stage, so that will be exosporas. Mostly the slime mold, they are endosporas, except three species of slime mold, other are mostly endosporas. Here in the picture we see the spore of Enteridium lycoperdon. Here we see that they have this membrane like structure. This membrane like structure, white membrane, it is called peridium or the cell wall. And this peridium, though it is called cell wall, it is non-cellular, and it has a various network like structure. Here we see that the tube like structure is there, and this tube like structure, it is called the peridium. This tube like structure, it is called the capillum. This entire bag like structure, it is the outer membrane, it is called the peridium. And the peridium, the tube like structure in the peridium is called the capillum. Here it is called the capillum, and in between the capillum, in the peridium layer, are salts of calcium embedded in the layer. So inside this sporangiophore are the microspores that generate either mixed amoeba or swarmers. So they are either mixed amoeba or they are the swarmers. So these are there is two phase of the spores. So the sporangia, the sporangium or the sporophore is of three types. One is the common one, that is called the sporangia, where it can be stalked. Here we see the stalk, or it can be unstalked. Here it can be sessile, but this base on which it is present, this base, it is the plasmodium. It is called the hypothalamus. This is the hypothalamus, and this type of sporangia, it is called the Acria physarum trichia. This is the most common, where it has a sporophore and a sporangial sac, or they are just sessile, and they can be united together. The sporangiophore, here we see that the stalk is united together, but the sac portion, it maintains separate identity. So this type of sporangia, this is called the sporangia. Then we see another type, that is called the Athalia. Here we see that the the sporangia, they are fused together such that this, say for example, this entire ball like structure, it is not a single sporangia, but many sporangia, they together they form this sac like structure, where entire many sporangia together, they they are covered with a single common layer, and individual sporangia do not have a individual membrane. So they have a common hypothalamus, and they have a common peridium. So there are many sporangia which are aggregated together, having a common peridium and common hypothalamus. So in picture we see that of the Lycogala. Another type of sporangia also present, that is called the plasmodiocarp. Here the sporangia is sessile, but it does not have the typical sporangia like structure. Here the sporangia, these typical sporangia like structures are absent. They are absent in the plasmodiocarp. Here the sporangia are in the form of venation. That is here. Here in this picture, if we see hemitrichia plasmodiocarp. Here this reticulate venation, they contains inside them spores inside them. So we see three type of sporangiophores in the slime mold. One is the sporangia, another is the Athalia, another is the plasmodiocarp. So now we see the life cycle. Here we see, if we see the amoeba, the mixed amoeba or the swarmers, they undergo fusion, and they undergo fusion to form diploid cells, and gradual they increase in size by division of repeated division of the nuclei and increase in size of the protoplasm, or by fusion of further amoeba to form the plasmodium. And then when the food is scarce, that it is amount of food is very little, then they form this sporocarp, that is haploid. And the sporocarp, they have a sporangiophore, and they have a sporangium. Inside the sporangium are the spores that are haploid. One thing I forgot to mention is that this plasmodium, they are diploid, but there is also another alternative method where and the amoeboid group, where the plasmodium is formed, but that is of haploid nature. They also form the plasmodium, and there is no reduction in the there is no meiosis in that of the. This was all about the life cycle of the slime molds, and In the next video, I will be discussing about general feature of fungi, and thereafter I will be starting several videos regarding the fungi. So, in case you have some queries, you write it in the comment section. Also, do share your comments in the comment section, and if you like the video, do share with your friends, and of course do subscribe my channel, because your subscription will encourage me. And thanks for watching this video.